This video will cover subtopic 2.7, DNA replication. The understandings, application, and skills will be related to that of DNA replication. So DNA replication is semi-conservative. When a cell prepares to divide, two strands of the double helix from the DNA will have to separate. Once those strands are separated, each strand is going to serve as the template for the new strand to be made. The way this works is that the base sequence on the template, the old strand, will determine the base sequence on the new strand. So when DNA has finished replicating and we have two molecules of DNA, each strand will contain one old strand and one new strand. And this process is referred to as being semi-conservative, being that this new DNA that's made contains one old strand, one new strand. The other DNA molecule contains one old strand, one new strand. And this works because adenine will only pair with thymine, and cytosine will only pair with guanine. So looking at this diagrammatically, if we have our DNA strand represented by the blue ribbon, and this is the DNA that is present currently in the cell. When the cell enters the S phase of the cell cycle and that DNA is synthesized, that old DNA or that current DNA will be used as a template to build a new DNA. So the two strands will have to separate, and then each strand will then be used as a template to build a new strand. And that new strand is represented by the red ribbon in this case. And the way that this can work and the way that each old strand works as a template, if we have a look at the bottom diagram, is that if the blue part of the diagram represents the old strand or the current DNA, the sequence of those bases will determine the sequence of bases on the strand that is being built or synthesized based on complementary base pairing. And the other strand will be built in the same way. So the base sequence on that other old strand will determine the sequence of DNA to be built on that new strand. So how do we know that DNA replication is semi-conservative? Well, when scientists were studying this procedure, or this process, there were three possible mechanisms of DNA replication that were put forward. There was conservative replication, in which the old DNA, as it underwent replication, and when that replication was finished, both strands from the old DNA would be conserved together, or they would be found on that one DNA molecule. And the new DNA that was synthesized was made up entirely of new DNA. So both strands were conserved in the old DNA. In that process of semi-conservative replication, as the DNA is being built or synthesized, the DNA molecule would contain one old strand of DNA and one new strand of DNA. So it would be a mix of the two strands. The last mechanism is the dispersive mechanism, and this is where when the DNA replicates, it is essentially a mix of both old and new DNA. So essentially, the strands get chopped up and reassembled, and you have this intermingling of new DNA and old DNA. So to test this out, an experiment was done by Messelson-Install, which is now known as the Messelson-Install experiment. Messelson-Install used bacteria that were grown on medium containing nitrogen-15, which is a rare isotope of nitrogen and heavier than nitrogen-14, which is the common kind of nitrogen. Nitrogen-14 makes up pretty much all of the nitrogen that we have here on Earth and is the most common form of nitrogen. What makes nitrogen-15 heavy is that in the nucleus of this particular element, there are 7 protons, and 8 neutrons, whereas nitrogen-14 would have 7 protons and 7 neutrons. That one extra neutron present in nitrogen-15 is what makes it heavier. So bacteria were grown in this medium containing only nitrogen-15 as their nitrogen source, and then grown for one generation in medium containing nitrogen-14. And when we say for one generation, we mean one round of cell division. So, in other words, one round of DNA synthesis. After this, the DNA was isolated and then centrifuged in cesium chloride solution. And with centrifugation, this is basically a process in which a solution in a test tube is spun around really fast. And then the components within that test tube, as it's being spun around, will start to migrate to the end of the test tube or migrate towards the bottom of the test tube. Things that are heavier will migrate further. Things that are lighter will stay closer to the top. So DNA containing nitrogen-15, which is a heavier isotope, would migrate further down that test tube, whereas DNA containing nitrogen-14, which is the lighter isotope, would stay closer to the top. So some of the results that Esselstyn and Stahl saw as we go through this. So in the first test tube, which is the control group, which was bacteria grown only in nitrogen-14, that lighter isotope caused the DNA to stay closer to the top of the test tube. When bacteria were grown in nitrogen-15 only containing medium, that DNA isolated and then centrifuged, that DNA was found closer to the bottom because nitrogen-15 is a heavier isotope and would have caused the DNA to migrate further down the test tube. When Esselstyn and Stahl then grew bacteria in nitrogen-15 for one generation, or grew them in nitrogen-15, and then transferred the bacteria to nitrogen-14 for one generation or one round of DNA replication, what they found was that there was essentially the DNA was essentially found in between where you would normally find the nitrogen-15, so it didn't migrate as far down the test tube, but it didn't stay as high up as the nitrogen-14 did, migrated somewhere in between. They performed another experiment where bacteria were again grown on nitrogen-15 containing medium, transferred to nitrogen-14 containing medium for 2 rounds of replication, so 2 rounds of DNA synthesis. And the result that they found there was that, again, they found DNA in that middle kind of area that they'd seen with just the 1 round of replication. But they also found a second band that was higher up found at a higher location than the other DNA band. And this was at a similar location to bacteria that were just grown in nitrogen-14 only medium. So these results suggested that DNA replication was semi-conservative. If we have a look at these results in terms of how they ruled out the different mechanisms, so if we have here bacteria that were grown in nitrogen-15 medium and then transferred to nitrogen-14 medium, again they found just 1 band of DNA. If replication of DNA was through conservative mechanism, we would have expected all of the nitrogen-15 DNA to be found together and all of the nitrogen-14 DNA to be found together. Remember, as the DNA is made, the new DNA stays together, the old DNA stays together, so they're separate. And then when that DNA would be centrifuged, we would expect to see 2 bands. But because we did not see 2 bands, it ruled out conservative replication. However, with semi-conservative replication, we would expect to see one band, and the same with dispersive replication. So after one round of replication, it was not possible to rule out dispersive or semi-conservative. So there were still two possibilities for a mechanism. This was settled by performing a second round of replication. So again, bacteria were grown in nitrogen-15 containing medium, transferred to nitrogen-14 containing medium for 2 rounds of replication. And what they saw here were 2 bands of DNA that were present. And comparing this, we'd already ruled out conservative replication. So comparing this to what would be expected in semi-conservative replication, because every time the DNA replicates, the new strand is found with the old strand, you would expect to find a strand containing nitrogen-15 and the DNA molecule that also contains nitrogen-14, the band here. And then as the molecule replicates, one of those original nitrogen-14 bands would have been used to build a nitrogen-14 strand. So you'd expect nitrogen-14 only DNA at that higher level, which is what was seen in semi-conservative replication. With dispersive replication, because you're always getting mixing of the 2 strands of DNA, you would only ever expect to get 1 band. Because 2 bands were seen, it could rule out dispersive mechanisms. And so semi-conservative was determined to be the mechanism through which DNA replicated. So one of the things you'd be expected to do is to be able to analyze the results that Vesselson and Stahl obtained in support of semi-conservative replication of DNA. And this we will cover in class. So looking at how DNA replication happens through the use of enzymes, there are 2 enzymes that you will have to know and how they, with DNA replication, or what their role is in DNA replication. The first enzyme involved in DNA replication is an enzyme called helicase. So helicase is an enzyme that consists of 6 globular polypeptides arranged in a donut shape. In this diagram here, helicase is represented by this wedge shape. So helicase, the function of it is that it will unwind the DNA molecule and will separate the strands of DNA by breaking the hydrogen bonds that hold those 2 strands together. So helicase will essentially move down the DNA molecule, separating the 2 strands, creating individual strands. And it will unwind the DNA as it separates those 2 strands as well. For helicase to perform its function, it does require energy in the form of ATP, and this is needed to move helicase along. Once the 2 strands have been separated, then the new strands can be created using the old strands as a template. This is accomplished by the enzyme DNA polymerase. So once those 2 strands are separated, the creation of the new strand is performed by that DNA polymerase. And the way that DNA polymerase will do that is that 3 nucleotides found in the nucleus. So you'll have free adenines, free thymines, free cytosines, free guanines just floating around in the nucleus. These will be collected by DNA polymerase and attached to the new strand by complementary base pairing. The DNA polymerase moves in a 5' to 3' direction. And the DNA polymerase will create a covalent bond between the phosphate group and the ribose. So it basically will attach those nucleotides together. So in our diagram here, if this what I've outlined is our template strand of DNA, the DNA polymerase will simply attach on to the DNA molecule and will start synthesizing that new strand. And as it moves along, so it will move in a 5' to 3' direction. As it moves along, it will start incorporating nucleotides or collecting those free nucleotides and incorporating them into that new strand. And it will use the bases on the template as a means of dictating which nucleotides need to get incorporated. It is also important to note that each template strand gets a different name. So one template strand is referred to as the leading strand. The other template strand is referred to as the lagging strand. Because DNA polymerase can only move in a 5' to 3' direction. On the leading strand, the DNA polymerase can simply move along following helicase, creating that new strand as the DNA is unwound. On the other strand, which we'll refer to as the lagging strand, because only portions, as portions get exposed, DNA polymerase can only move in that 5' to 3' direction. So synthesis is always like ongoing or having to start again. The DNA polymerase always has to reattach the newly exposed sites of that template strand and then start synthesizing those portions. So that's why it's referred to as the lagging strand. It's not a continuous process, it's actually discontinuous. DNA polymerase will also function to proofread the complementary base pairing. So if there are nucleotides that get incorporated that shouldn't, the DNA polymerase will realize that mistake and then edit and put in the correct base. So it ensures that there is conservation of that DNA sequence. The other thing that I'll mention about the DNA synthesis is that in order for the DNA polymerase to attach onto the template strand and start synthesizing that new strand, there has to be a small segment of DNA that is already attached. That small segment of DNA is referred to as a primer, and you can think of it as that small segment of nucleotides that will prime the DNA polymerase to start synthesizing. So ultimately, these steps in DNA replication can be used by scientists now in a process that's referred to as the polymerase chain reaction, or PCR for short. So in this process of PCR, this can be used when small samples of DNA are obtained and we need to, or scientists need to now create a larger amount of DNA in order to sequence it or determine what DNA is present. So in PCR, the DNA sequence gets denatured or basically separated at a high temperature. Remember, high temperatures can break hydrogen bonds, so increased temperatures can cause DNA to separate at high temperatures. The temperature then gets lowered within the reaction vessel that will allow those primers to bind. And remember, those primers are short DNA sequences, which in this case will be short DNA sequences that are complementary to the region of DNA that we're interested in amplifying. So the temperatures are lowered to allow those primers to bind, and then the temperature gets raised again to allow DNA replication to occur by an enzyme referred to as Taq polymerase. So what is Taq polymerase? Well, Taq polymerase is a DNA polymerase obtained from a bacteria referred to as Thermus aquaticus, which is a bacteria that's found in hot springs. The Taq polymerase is a stable enzyme at high temperatures that would cause DNA to denature. So that enzyme can withstand high temperatures and still function properly, whereas a DNA polymerase from a eukaryotic cell, for example, would not be able to withstand those high temperatures and would not be able to function. So the way this looks in terms of a graph is that you have your temperature set at a relatively normal temperature, lower temperature, which gets increased to almost boiling levels. This will cause the DNA to split and then separate. The temperature is then lowered to allow the primers to bind to the interested regions of DNA, and then the temperature gets raised to allow Taq polymerase to now create that new sequence of DNA. And then when that new sequence of DNA is then created, the process is then repeated. So the temperatures get increased to now split that DNA. Temperature gets lowered to allow those primers to bind. Temperature gets raised to allow Taq polymerase to do its thing. This process gets repeated anywhere from 20 to 25 times to allow that DNA to be amplified in quantity. And then once that DNA is amplified in quantity, it can then be sequenced or then can be visualized by a process called gel electrophoresis. So that is the end of this video for 2.7 DNA replication.